Hello, my name is Ivan Kolutza and I lead the computational biophysics group at SIC Biomaguna in San Sebastian. Today I will talk about a methodology to classify the evolutionary importance of residues on protein structures. Typically, residues on a protein are classified according to their relevance in the particular property under study. Stability, catalysis, for example, protein-protein interaction, or allosteric transitions. A powerful tool to characterize amino acid function or relevance is through multiple sequence alignments, where residues are classified according to their conservation along time evolution and their pair correlation, also known as coevolution patterns. Overall, we can classify residues as conserved or co-evolving hence. Now, the objective of this talk is to further rank such a classification according to how functional or structural each residue is. By functional, we refer to the importance that the particular residue has in the protein function, while by structural, we refer to how important a residue is for the stability of the folding. Of course, residue could play a double role. The first step is to generate the ranking is to extract residue coupling strength from the coevolution data. The reason is that we need to filter out indirect interaction and focus on direct couplings. For that, we use the direct coupling analysis, or DCA. DCA has been proven to be extremely effective in predicting contacts between amino acids directly from the multiple sequence alignment data. In fact, the very recent result on protein structure prediction using deep learning, for example, the AlphaFold of Google and the new Rosetta implementation from the group of Eric Baker, directly sprouted from the DCA methodology. Deep learning extends the mean field approach of DCA to a multidimensional analysis on which neural networks are uniquely suited for. But this is a very interesting field of discussion, but there is no time to talk about it right now, and I would be more than happy to discuss about it later on. So here, for example, there are two systems that we directly apply the DCA for. For instance, the first one is this protein system which has two domains, an ISCA domain and an ATPase domain, and you can use DCA not only to predict the contact between the residues within the domain, but you can also predict how these two domains should arrange in their quaternary structure within the same protein. You can also extend this to predict how two proteins interact, or better, two domains from two different proteins interact, and predict which residues should participate in the binding. Now, I have to say that this prediction is much tougher at the moment, and one of the reasons is because it's not easy and trivial to produce the correct multiple sequence alignment when you have pair of different proteins. And this is, again, it's a different topic that would deserve a talk of its own, and again, it's a fascinating subject that we are working on at the moment. Now, what we thought at this stage was, okay, we have this very powerful method, the DCA, and what happens if we use the DCA to predict the contacts or the key residues on a computer design multiple sequence alignment? In other words, if you produce a lot of artificial sequences and you align them for a single protein, of course, what would the DCA predict? Now, we have to start from an hypothesis that your sequences that you produce with your artificial methods must all fall to the target structure, because otherwise you will generate noise, or you are introducing noise in your multiple sequence alignment. But provided that's guaranteed, what we expect is that the DCA will predict contacts or high correlation direct couplings between residues that are key only for the structural aspect of the protein, in other words, for its folding. That's because during the design, unless you purposely do so, there is no selective pressure towards functionality. So we thought what if then we basically apply the DCA to artificial sequences and natural sequences, and then we compare or subtract one another, obtaining basically a classification that it's just a ranking of residue according to how important they are in an artificial sequence compared to how important they are in a natural system. And that's why we call this methodology the differential protein annotation method. Now the first step is then to basically choose a model that produces such artificial sequences that are guaranteed to fold into a protein structure. And bear in mind that your protein structure space must be comparable to the one of real proteins, because you want to capture correlations between residues that give you a correct folded structure comparable to what natural sequences do. So we chose a model called the caterpillar model, which we developed already some years ago, that we extensively tested this model, and one of the properties that it has that fits perfectly with our needs is exactly to produce sequences, completely artificial sequences, that do fold to realistic protein structure. And in fact, it is particularly suited for this application because the caterpillar sequences have little resemblance to natural sequences, and indeed they are only designed to fold to the target structure. So there is no risk of accidentally implementing any functional information that would arise, for instance, by taking natural sequences and mixing them with the artificial ones. So in fact, you can prove that the sequences generated with the caterpillar model have a global free energy minimum in the target structure, and target structures that are taken directly from the protein data bank. So these are proteins exactly as you would find them in nature, and the artificial sequences, for instance, I list them some below here, fold back to those exact structures. And the list of sequences goes on and on. So what we have now is a multiple sequence alignment of the natural sequences, and now we can produce an artificial one. We apply the DCA on both of them. We look for the highly conserved residue, and what we get is that, for instance, is a ranking like this one, where we applied it to the FKBP12 protein, which is the FK506 binding protein 12, where again FK506 is an important immunodepressant used in organ transplant. And we chose this protein because, apart from its importance, the FKBP family is a highly studied protein family, so there are a lot of examples to generate an extensive sequence alignment database, which is an important element for using this method. Now we took the same protein, we run it in the caterpillar, we designed it to produce a set of sequences, and from the comparison of the DCA analysis, we can classify residue according to how structural they are in orange, how functional they are in green, and how intermediate they are. In other words, that they have a similar weight in functional and in natural and artificial sequences, in that we plot them in yellow in this case. I have to say that in blue, instead, we plot the residues that are being experimentally identified to be the one in the binding pocket for the FK506 molecule. And these are the ones we also find perfectly aligned with our prediction of the green functional residue. So this is an example of a fully successful prediction of DPA, where all the functional residues correspond to something that has been identified in nature. Now, this is not always the case, unfortunately, because the model has limitation due to the noise coming from the multiple sequence alignment, the problems in the sampling of the sequence space with the caterpillar models, all things that we are still working on to solve. And in fact, in some cases, the prediction is much worse, with, in this case, is the a protein from the PDZ protein family, another very large protein family that, on the other hand, has a huge spectrum of functionalities. And in this case, we have only 61% of the functional residue identified in with our method. And you can see in red, we show the one that they are experimentally known to be functional residues that we do not capture. And of course, this could depend from a lot of factors. We believe that mostly is because of the way the alignment data has been grouped together, because it does take mix together different functions from different organisms. So we might want to extend our methodology and separate it by subclasses to see how the method would improve. Now, another thing we tried was to compare what happens if you replace the Rosetta design method, the Caterpillar design method, with a more established method, which is protein Rosetta. So we apply this to the CTPR8, which is a 8 repeat of the same protein, and we designed the core of this protein. As you can see, the percentage of predicted residues that are known to be structurally important in this case is in yellow here, are highlighted from literature, and the one from the Caterpillar in purple and in from Rosetta in green, the percentage is comparable. But what Rosetta does is to make a very extensive list of highly conserved residue, and this is because Rosetta has normally a very well defined global minimum for the protein design with very small variability, probably coming from the minimization algorithm that it has. We are now working to try to force Rosetta to produce a larger spectrum of sequences. So with that, I would like to conclude with a couple of take-home messages of DEPA, and say that DEPA can efficiently predict protein functional annotation, residue annotation, without any prior knowledge of biological function and directly from multiple sequence alignment. It is a promising technique that needs further refinement, in particular in the way the multiple sequence alignment data is collected, and as well a better sampling algorithm of the design to efficiently produce a large collection of solution. I would like to thank the invaluable collaborators that worked with me during the years and currently working on this project, and the funding agency and you for your attention. Thank you very much.